Thank you for nice introduction. I used to be in a position to invite keynote speakers for LNF Symposium when I was served as a director. But it was a nice revenge way to invite me as a keynote speaker to the symposium. But I'm gladly to be here and talking about some of my research, especially for this gathering, sharing all the knowledge and expertise that we learn, and also talk to many users, and so that we can learn from each other. And for the next hour, I wanted to introduce some of my research that I do, how engineering can help and contribute to advance the science and medicine. And before moving on to this topic, I cannot resist to show this slide. When I was serving as a director of the LNF, and Ann Ludy actually visited us in one of our review sessions. And after she took a look at the facility and to get the presentation of what we do with her money, and then she generously gave a $1 million check to us. And this is the picture we took at the time. And Dennis Grimard was the managing director. Sandrine is not here because she took a picture at the time. So she's still here. But I just wanted to show this slide because we would like to thank all the contribution and donation from our alumni and the contributor to this facility so that we can do a lot of good research. I'm actually asking this question: what are the important problems that we wanted to attack and solve as an engineer? There are two. When I take a look at the research that I do in my laboratory, one is related with the cancer. Cancer is the disease that still need to be overcome. And we have a battle to really fight against the cancer. A lot of people suffer from this disease. And still there are a lot of opportunity for engineering can help to medicine to help all the patients and then move the cancer biology to understand more about the cancer. The other one is the brain. It's the last frontier to explore. There are still big mystery between your 2 ears, that is your brain. Still we don't understand how it works, even though you live with that. So now there are a lot of newer technologies that are being developed to tap onto the brain activity and try to understand. If we understand how brain works, then we may be able to find a treatment for these debilitating brain disorders. So let me start with the cancer. Maybe you have seen this slide many times, lab on chip, and the basic concept is try to build a big laboratory on a small microfluidic chip or small silicon chip. And here the reason is, in all the activity here done by the researchers here in the laboratory, in a few days they're mixing the chemicals and reagent, and sometimes they culture cells and take a look at the microscope. But if you can integrate all this into the small microfluidic chip, and it can be smaller, so you don't need this lab space, and also it will be much faster. The experiment, which will take probably a few days, can be done in a few hours or a few minutes in a microfluidic chip. And also consumption of the reagent is very small, which is typically very expensive, so it can be low cost and everything can be automated. So people try to make all these petri dishes and multi-well plate into the small microfluidic platform technology. However, when you take a look at the microfluidic chip out there, is pretty outstanding. It's a fascinating chips. Even you can make microfluidic logic and microfluidic memory and microfluidic AD converter, and you can handle the cells and reagent into the multiple microwells. But if you wanted to really use it, this is the reality. You have to have a lot of connection. This is the liquid connection in pneumatic control and handling. It is pretty messy, and it is very difficult to operate, especially for the hands of the biology people or medical community. So what we have come up with is we wanted to develop a microfluidic chip which can be disseminated to the community and science community so that they can use it very easily. So our approach is user-friendly microfluidic chip. So we get rid of mostly other very complicated control mechanism or external hardware so that low expertise are required and any technician can handle this at high throughput with the minimum overhead and by using very simple pipette operation rather than complicated control mechanism. And also we wanted to make it compatible with the existing current biology protocols so that they can apply all the experiment that they do in the Petri dish on this microfluidic platform. And this is a very simple structure, and the array of microwells and the size of 100 microns. We have a multiple inlets, and the one inlet is loading cells. In this one, we try to look at the single cell because there are heterogeneity of cancer that makes the cancer treatment very difficult. So we wanted to understand how each cell is behaving differently. So when you load the cells, we are capturing cell in each microwell, either single cell or multiple cells depending on the configuration inside a microfluidic chip. And once we load the cells, we are loading a reagent or drugs into the different port so that we can have multiple assays done at the same time at the high throughput. This is the initial device that we come up with, a very small number of arrays, 64 microwells. Each microwell has a interesting structure inside, which we call the capture site. And here we have actually providing two flow paths. One is the center path with a shortcut, and the other one is a path like a detour, which has a large flow resistance. Therefore, when the cell is loaded into this microfluidic chip, it will take a shortcut, the center path, coming into this capture site and the capture here, and blocking this path. Once this shortcut is blocked, the cells coming later will take the detour in taking and then coming to the next chamber and capture in the next chamber. You can provide a multiple site and capture multiple cells, pairing cells and 3 cells or 5 cells. Once we capture the cell, we can culture them, and also we can inject the reagent or drug from the multiple port for high throughput assays together. The next slide shows you how it can be done here. This is the illustration. We are using a microbead for the visualization purpose, but it can be done with the cells very easily. So when the cell is being introduced, it's coming into the center and readily captured at the center. And then you can move on to the next chamber and capture here in the middle, and then the uncaptured cell will be coming into the next chamber and captured in the sequentially in the next chambers. So within few minutes, without doing anything, this once you're having a pipette operation, loading cells through the gravity flow, cells are being flowed into the microchamber and deployed as single cell in a multiple way. So having this, we are developing single cell assay microsystems, integrating all these wells, and we culture cancer. So we just having a cancer on chip, and for the cancer drug screening, and study cancer cell differentiation and stem cell culture. So let me introduce a few platform technologies that we have developed. Once we load the cells, a single cell is showing here, and each microwell has a single cell with a very high capture rate, over 80 to 90%. And this is the prostate cancer cell. Once we culture them, it has a clonal culture, which means in the population of each microwell coming from the same progenitor cells, same mother cells. So there are same type of cells. But we found that there are three different phenotypes of the prostate cancers, halo and melachrone and parachrones. Their morphology is different, and also when we treat the drug, docetaxel, that's the D drug for treating prostate cancer, we can see a different drug efficacy. That means that each patient may have a different phenotype composition or different types. Then a drug may not work effectively or may work at only one specific phenotypes. So this can be done by taking a look at the single cell resolution and tease out what kind of cell types the patient has and what kind of drug has to be treated. So we study this in more and expanding our microfluidic chip into the 1,000 channels. And again here, no external pump and no external control instrument are needed. And then and the behavior of the population and behavior in a single cell resolution can be analyzed. And we can load the cells, and as you can see, we can distinguish the one cell from another. This is staining. Once we can do that, what we do is we can develop we can help to developing a drug targeting cancer stem cells. Actually, there is a cancer. Cancer cells are not all same. There are cancer stem cells, which is considered to be a initiation of the cancer, and also coupled for metastasis of the cancer. So it's important to really treating these cancer stem cells. When you take a drug, it may kill only the bulk tumors, but does not kill cancer stem cells, then cancer will relapse. Once it comes back and there is a mutation, it will become a different cancer, so treatment will be difficult. So by utilizing our chip, we can load it into a single cell. We can identify which cell is a cancer stem cell, and then we are target drug for that. This is one of the examples that we have done. This is a control system plan. And this ALDH positive is the indication of the cancer stem cell, and negative is the non-cancer stem cell, which is the bulk tumors. And this control drug can effectively killing bulk tumors, but not the cancer stem cells. So once you treat the drug, then tumor may shrink, but it doesn't kill cancer stem cells. So there's a high possibility that the cancer will relapse. But these are the other drug developed from my collaborator lab, Vukanovich lab. And this drug effectively killing cancer stem cells, although not killing bulk tumors. So there's evidence that we have to have a cocktail of drugs which not only treating bulk tumors, but also killing cancer stem cells. So what we do here is once we treat the drug, and then some cells doesn't really respond to the drug, which we call drug-resistant cells. And then we wanted to take it out the cell and then wanted to see the gene profiling so that which gene we have to target for those cells. So we developed the microfluidic chip in which each target cell can be retrieved, harvested, so that we can do a downstream analysis, for example, gene sequencing. So here, we just having a little bit modification of the microfluidic chip and putting a very thin layer of carbon nanotube and replaced by other thin layer of the metal layer. And having that, and we have a nanosecond pulse laser shining upon the target cell, and then this laser energy will be absorbed by this metal layer, and then there is the temperature increase and the buckle this microfluidic chip, the PDMS layer, and detach the cell. It's amazing, and it works well. This mechanical detachment can target for single cell in each microwell and detach the cell without damaging them. So we can retrieve the cell live. So this is the movie that we capture. There's a single cell in the middle, and then when we shine the laser, nanoperson that we can detach the cell, and we can harvest from this microfluidic chip for downstream analysis. So we retrieve the cells being cultured and compare with the gold standard of the typical detachment of the cells. It's a trypsinization, and this is a chemical and being used for this biology studies. And once you compare with this trypsinized cells with the laser detachment cell, there's not much difference in the cell viability. That means that the laser detached cells has a pretty good viability in healthy cells. And also we did gene profiling, whether this laser detachment will affect some of the gene expression. We don't see much difference between trypsinized cells and laser detached cell. So this one is indicating that all this single cell detachment from this laser technique allow us to having gene expression profiling of the target cells. So currently what we do is having all this assays completed. We're taking the cells and take a look at the gene profiling. And cancer can be managed if it doesn't do the metastasis. So people people don't die from cancer, but when the metastasis happen, and this is really the problem. So we developed a microfluidic platform to address, to answer the question, what mechanism is really playing a role in this metastasis and how do we block them? Metastasis is a multiple step process, and what is being known is when the cancer and the tumor is initiated, and then one cell, which is we call that cancer stem cell, a very motile cell, and gets into the blood vessels through the intravasation and becoming a circulating tumor cell, CTC. And then it finds a new home through the extravasation and getting into the epithelial cells and getting into the other region of the body that become a new cancer. There are multiple processes, and this one we can characterize the cells by migration chip and taking a look at the motility of the cells. And not all the healthy and somatic cells cannot survive in suspension. Cells wanted to be attached and together with the other cells. Only the cancer in the stem cell, which is induced this metastasis, can survive in suspension. So we developed the microfluidic chip to culture cells in suspension. And also the other one is cell-to-cell interaction. Some cells coming into the different region of the body and some cells don't. And the cell-to-cell interaction is really important to find a new home. So we developed a few of these microfluidic platforms, but let me introduce in a couple of them in the next few minutes. So this is the migration chip or motility investigation testing chip. And it is made out of from multiple narrow channels, and there's an inlet and there is the channel where the cells are being introduced. And I call this a cell raise chip, and cells are being raised through this microfluidic channel and coming into the other side of the channel. Once we having a different nutrient and growth factors in the other side of channel, cell wanted to get rich into that, they're migrating through this. And that is what we call the chemotoxicity and chemoattraction. But what we do here is, rather than having multiple cells loaded into this, we are loading each individual cell in each microchannel so that all the cells are initiating from the same point, rather than some cells at the entrance, some cells are in the lag, in the behind. So it does have a good profile of each single cell behavior. Also, we can have a pretty good statistical data out from all the cell behavior. This is the microfluidic chip. Initially, we are lining up the cells into the inlet of the micro channel. And after 1 day, cells are moving through this channel. And highly motile cells are moving much faster, and less motile cells, some lazy cells, and maybe wanted to stay there in this initial position. And when we're loading HGF, one of the growth factor, cell like them. And a lot of cells moving on to the other side. So initially, well, cells are loading as a round shape. After 1 day culture, they are attached and they start to moving through the channel. And this is the showing that how cells are crawling to this narrow channel, moving on to the next. And when we have increase the concentration of the HGF in the other channel, and obviously there's more cells moving on to the next cell. And this is the good indication of the heterogeneity of the assays and different cells and the different types of the SCOBY3 and other types of cells. One is stained green and red. These are all ovarian cancer cells. Obviously, you can see green cells are more motile, very aggressive cancer cell, while the red cells are pretty slow and less motile cells. And question here is, are there any difference between these two groups? So we studied that. So after migration, and we harvest them, and from the channel that we loaded, the cells are residing here is a lazy cell. They don't want it to move. And the cells migrate into the other channel as a very motile cell, a very aggressive cell, and we just harvest them. We see the morphology. Amazing enough, there's an obvious difference between the morphology. Non-motile cells are in a round shape, and highly motile cells are more elongated shape. Even when you take a look at this, it looks like this cell may move much faster. And we harvest this cell, and we just having a control, and we put it back into the mouse, whether there's any difference between forming a cancer from these cell groups. Obviously, the motile cells are making more cancer cells, and also the weight of the tumor is much larger than non-motile cells, and indicating that our microfluidic chip can screening much more aggressive cells. And also, in order to take a look at the metastatic nature of the cells, we just putting these cells back into the blood vessel of the mouse, and then it generating cancer in the different region of the body. And again here, high motile cells forming more cancers than non-motile cells. Currently, what we do is we are harvesting cells and then doing the gene expression study, which gene really triggering this high motile and migration behavior. Once we identify that, we can knock down this gene to suppress this metastasis. So one of the collaborator lab interested in one of the genes called P38 gamma, which is considered to be the gene would turn on motility. So in order to study that, we just making a little variation of the microfluidic and putting a chalk and change the size of the chalk and to emulate the capillary. We studied two cells, one the wild cells and the other one is the P38 gamma knockdown gene. We see pretty different morphology from this. And what we have found is between these two cell groups, when this chalk is less than 6 micron, the knockdown gene really cannot penetrate this. There's a significant difference in the motility. So, and this microfluidic chip can screen and also validate which gene, and by having this knockdown experiment, and effectively suppress the motility of the cancer cells. And the other work that we do is we change the surface of the microfluidic chip, coating polyhema. It's the hydrogel, thank you, and coating onto the surface of the microfluidic chip. Once you have this this layer in the channel, It just observes the water molecule and prevent the cell adhesion. So we selectively coating this polyhema onto this microfluidic chip, making a suspension culture. So same microfluidic chip, but only the surface is modified. A cell cannot adhere onto the surface. And from the single cell, in making a sphere, because cells are kind of aggregated, and then making a bigger sphere. Only the cancer stem cell can make the spheres. This is one of the marker-free screening of the cancer stem cells. And the other cells can survive on this suspension culture. They're gonna die. So we do a lot of the drug assays by using this. And now we expanded our microfluidic chip from 1,000, 3,000, even now 10,000 microwells, and loading into the chamber and culture them and growing them and having a drug assay and the screening which drug is effectively blocking this sphere formation and targeting cancer stem cells. So I just briefly introduced a few microfluidic platforms to understand how cancer cell behavior and understand the heterogeneity and gene expression, how to suppress its motility and the stemness to treat the drugs. And I collaborate a lot of great oncology doctors in the cancer center, and there's a list of the people here, especially Max Wicha and Ron Vakanovich, who has been a great collaborator for over the years, and many students and former students and postdocs. Especially, I wanted to acknowledge the contribution of two previous students, Yuji Chen and Patrick Ingram. And let me switch my gear a little bit in talking about technology that we've been developed to interface with the brain, to understand the brain activity. And this slide shows you a snapshot of the history of the neural interface technology over the many years. And in late 19th centuries, Hans Berger started to tap onto the brain activity, putting the electrode on the scalp of the head and measuring electromagnetic wave and correlate with the brain activity. And he's a physician, he's not a physicist. So he studied this from his son, putting the electrode in his son and then monitoring the activities of the signals and correlate with the brain activity. And there are a lot of things that can be done. It's a very non-invasive approach. Still, a lot of EEG is being practiced in the medicine. But it's like, the spatial resolution is pretty limited. But it is like you just put the microphone outside the stadium and try to understand what's going on inside the stadium. There's a proof of play is being done, and there's a touchdown you may see. The crowd is like shouting and applaud. You may, but if the crowd is silent, you may guess that there's not much happening inside the stadium. So you can understand what's going on, but not exactly. So people wanted to get inside the brain to tap onto each neuron's activity. One of the approaches is ECOG, that is open the skull and remove the skull and putting the electrode on the top of the cortex. It has a better resolution. It's like putting the microphone inside a stadium, but still you don't understand what's going on, each individual person's behavior, each individual neurons. And after that, people putting the microwire into the brain, and pretty invasive, and try to record the single cell activities. In 1960s, silicon micromachining technology has been applied and bring the multiple electrode, pioneered by Ken Wise while he was at Stanford University. And that's the beginning of the Michigan probe. And once he moved to Michigan, and there are a series of Michigan probe has been developed from single shank to multiple shank, and stacking them into the 3D array and putting a electronic interface circuitry and accessing a lot of cells simultaneously together. And there are many other Michigan-style probe has been developed, including Freiburg and IMAG and also IME. There are other approach done here in the Utah and what we call the Utah, the probes in Utah array. And this is mostly targeting for motor cortex, human prosthetic applications, and FDA approved. And the question is, how many neurons can we access all together simultaneously? This is a plot. It is called neuroscience Moore's law. And you're familiar with the Moore's law. Every memory density doubles in 1.5 years. The neuroscience Moore's law is how many neurons you can access simultaneously. And it's pretty slow, 7 years, and then double the density of the neuron activity. In 1960s, maybe 1 or 2 neurons you can monitor together. Now it is like 100. I just plotted a few, the publication recently. Now we are reaching up to 1,000 channels. But remember, this is exponential. So within the next decade, we can reach up to tens of thousands neurons or 100,000 neurons, even millions of the neurons. And maybe this slope can change, maybe even steeper due to the technology evolution. And this is a slide that I borrowed from Ken Wise. And this is the Michigan approach. We wanted to have a penetrating silicon probe into the brain and to access a lot of activity of the single neurons and monitoring extracellular potential. And putting but this is a really widening problem. If the channel is more than 1,000, you cannot get the 1,000 wire coming out from the brain. This is nonsense almost. So we wanted to amplify the signal, which is very small, and digitize them and multiplex them. So reduce the number of wires. And this can be further processed digitally. Maybe data compression has to be done. And also signal can be transferred wirelessly eventually. This is Michigan architecture, and it's been working for many years. And this is the device that's been developed by Ken over the many decades, not just the years. This is the passive probes, and which means they electrode onto the silicon micro machine needle, no electronics, and each individual site can be accessed from the bonding pad from the body part of this probe. And this technology has been commercialized by NeuroNexus. You can buy one of these. And this technology has been evolved in this way, passive to active probes, because in order to access multiple channels, we wanted to amplify them, providing the circuitry for monolithic integration and hybrid integration. Now, Michigan approach is hybrid integration, take advantage of the all foundry service, having all the circuit designed and then fabricated by foundry and then integrate them together in hybrid packaging. And from acute to chronic, and then providing a very compact packaging. A lot of packaging effort has been done here in Michigan to make it small form factor. And the other one is heterogeneous integration. I'm going to talk about it a little bit. Not only for electrical recoding and electrical stimulation, and we wanted to have optical stimulation capability by integrating optical component onto these probes. So this is one of the approach that we've been gone through, and making a silicon probes here and attaching and package with the circuit. So circuit chip can be integrated in many different ways onto the silicon probe and minimize the number of interconnection lines from the multiple channels. This is one example, assuming that we have 128 channels of the recording, and then minimizing the interconnection, having the integrated circuit packaged in hybrid and assembled into the silicon platform and accessing multiple channels. And if we assemble 8 of them, there will be 1,000 channels. And this one illustrates that we need to develop the low power and a smaller area circuit integration. So this is the technology direction. We are developing neural interface circuits to minimize the power and also area together. This is a 16 channel that we developed, and that's what I just showed in the previous slide. And moving on to 128 channels. Now we have a 256 channel chip in a very small form factor in extremely low power. And this chip and having a amplifier and also a gain adjustment, AD conversion, and multiplexing to reduce the number of wires coming out. Especially in this configuration, we try to minimize the power and also area. We're taking advantage of the spectrum of the neurosignals. When you plot the spectrum of the neurosignals, it has a 1/f profile. So high energy in the low frequency range and also low energy in the high frequency domain. If you're an electric engineer, when you take a look at it and you may get some idea, you can utilize it, apply the delta modulation, and then reduce this dynamic range significantly, then you can release the burden of this amplifier and AD converter very significantly, implement the circuit in a low power in small area. Once you process all this, and all the digitized sigma delta modulated signal can be retrieved by sigma modulation, which is integration, then retrieve the whole signal. And this works beautifully well, and then we can implement a circuit in a very low power and a low consumption of the area. And this 128 channel chip works pretty well for multi-channel recording from the mouse, all simultaneously together with a small power consumption. So we are continuously pushing this direction to integrating all of the circuitry toward the 1,000 channel and beyond. For the remaining time, let me talk about the other stuff, the final topic of the optogenetics and lighting the brain. And neuroscientist community was excited by the new possibility of the optical stimulation. When you do the electric stimulation, all the cells within the 100 microns away from the stimulation site will be all stimulated unselectively. If you have optogenetics and there's an opsin that can be transfected into the target neuron, when you're signing up on the specific wavelengths, for example, blue, only the target neurons can be stimulated and excited. That provides a tremendous opportunity to understand local circuit connectivity in the brain. But most of the work done in the neuroscience community is sticking to the optical fiber into the brain and modulate brain activity, shining the light onto the whole region of the brain. When I take a look at it, I think readily MEMS technology can help this to deliver the light locally into the target region of the brain to understand circuit connectivity. So when I arrived here, I just tried to attack this problem by integrating optical waveguide onto the Michigan probe. And initially, we started with the polymer waveguide directly integrated onto the silicon probe and optical fiber coming into. We can etch the silicon, the micromachining. We can self-align the optical fiber to the waveguide, and then we can deliver the light. And the light is coming up from the distal end of the optical fiber. The beauty of this is the monolithic integration. So we can control and the location of the illumination site respect to all the recording channels. Later, we changed the dielectric waveguide to the polymer waveguide to dielectric waveguide for a better reliability. Also, we can reduce the intrusion of the tissue and scaling the size from 15 to 5 micron and improve the efficiency much higher than polymer optical waveguide. That works pretty well. And the question here is, can we deliver different colors to the same distal end by switching the light source so that we can either excite the neuron by shining the blue and inhibit the neuron by shining the yellow, and each different opsins, channel rhodopsin responding to blue and halo rhodopsin responding to yellow. So we can have a freedom to either excite and inhibit the neurons by switching the light. we adapt an optical mixer technology and embed it into the silicon, and then we should be able to switch the light onto the silicon probe. Later, we just eliminate this optical fiber because still the big optical fiber has to coming from the light source into the probe, and the big tethering problem, it's not scalable. So we integrate the light source in the back, which is the laser diode. And one of my students come up with a brilliant idea how to integrate in the small form factor. So there's no optical fiber connected here. When you take a look inside, there's a green lens here. And the LD is coupled to the green lens and providing a tremendous amount of the misalignment tolerance. You can deliver light into the optical waveguide and switching the LD simply by electrically. And then you can switch the light wavelength in the distal end. So this is how it works. When we switch on the LD, we can shining the either blue or red or together. By having this, we have a pretty good alignment tolerance, so we can expand it into the multiple shank and the multiple configuration. And this one have a 4 shank and 8 LDs are integrated and set-aligned and through the green lens, deliver the light to the distal end. And showing here at the end, and we have the light coming from by only turning on and off the LEDs in the back here and switching these LEDs electronically, and then providing the light, either blue or red, so that we can stimulate the different neurons. Having this, what we can do is actually, by blue, we can excite the neurons. And by shining the red, we can inhibit the neural activity. This one shows you that one of the evidence we excite the neuronal activity and we can suppress it. Also, we can tease out some of the other connectivity of the circuit and pyramidal neurons and interneurons. And this is one of the evidence that there's a closed loop feedback. And once we increase the intensity of the red, and we can suppress the activity of the excitation of the pyramidal neurons. This is indicating that interneurons are having negative feedback of this pyramidal neuron. So this one opened up the possibility to understand all this circuit connectivity in the each video neurons at single cell resolution in a very complicated and complex, highly densely populated region of the brain, which is the hippocampus, where the memory is being formed and then retrieved. But still, we have a light source outside the probe shank. The question here is, can we integrate a light source directly onto the probe shank? The answer is yes, we can do that. So we integrated micro LED in the neuronal size, about the 10 micron, onto the probe shank directly by utilizing gallium nitride on silicon wafer. This one can provide a really high density, the array of the micro LEDs onto the shank, and we can turn it on and turn off all these LEDs by only electrical signals. This is possible because the actually LED lighting, the technology, a lot of technology has been invested. Now growing gallium nitride on silicon is become a commodity. It was a dream in 1990s, but it's now commodity. So we can buy the wafers. So we buy the wafer, gallium nitride quantum well grown onto the silicon. We etch and making a MESA micro LED, and then we integrate all this electrode and all this neurotechnology, and then MEMS. We etch the silicon. We know how to do that. So we etch the silicon, making a needle structure. Same Michigan probe, but we have additionally having LEDs on top of that. So this is a multi-shank configuration, and this one has 8 recording channels, but 3 LEDs in each. And we have a pretty well-defined micro LED structure on each shank. When you turn it on, this is the probe on the US penny. You can turn on each LED and one of the LED, or all of them from each shank. If we can do this, what we can do is this, and this is a little bit busy slide, but just bear with me for a while. This is the series of the experiment done in Euisik lab. He's one of the great guy who I enjoy collaborate with. And here, this LED is on, and these are the circle indicating the neuronal response, and with the different phase. Red is the 0 phase delay, which means that this is a direct excitation from the LED light here. And the other neuron here is a phase delay, is an indirect excitation. By shining upon the different LEDs sequentially, you can understand which neurons are responding directly or which neurons are responding indirectly, and configure all the connection in the very densely populated neuronal in the region of the brain, a single cell resolution. And this can be done because you can eliminate very, very small power, only eliminating and exciting neurons in the vicinity of the LED down to the single cell level resolution. And this is the way we eliminate the light, and my students record this with the music. I'm not sure whether you can hear the music or not. And this one has a freedom to eliminate any LED, because you now see the LED TV, so you can selectively excite neuron activity and region of the brain, and also you can record how this excited neuron signal is propagating through the complex network of the neurons. With that, I'd like to thank the collaborator, and Ken was a great guy, and still he's active, although retired, and working with me. And John Samo is I joined in a group and with the industry experience, really pushing this technology a lot. And also Yuri Basaki is a neuroscientist in New York University. Without him, we couldn't do all this in vivo experiment done, and cannot validate our technology. Many the students here and postdocs, and I wanted to acknowledge two, Fanu, who did the micro-LED work, and Komal Kampasi, who did for waveguide probes, and the funding agency. Most of all, Lurin Nanofabrication Facility. Without this facility, I could not make any of the devices that I showed you today. I really appreciate all the great facility, and also, most of all, the staff. The facility doesn't operate by itself. With the staff and with the help, and I really appreciate all of your contribution to allow us to do the prime research altogether. With that, I'd like to thank you for your attention. These are the group of my students work together. Thank you very much.